Hello students, I am making this video to support Lab 1 in environmental science. There were lots of people who collected data well, like for example this is a student collected table of data for the lab, but a lot of people didn't get to the place where they made a graph, and so I was set out to make a graph for you in which I manipulated the data in Excel. But I have lots of videos like that in a playlist I'll post on announcements. And I lost my computer, so I've been using my phone more. So right now I'm using my phone, and I just discovered that your phone can do something truly amazing, which will help you learn. So here, look, here's some data, right? Okay, we've got good metric units up there, and we also have that the units are not actually in the cells. We just have numbers in the cells, okay? So now we've got some other data over here. Let's just look at this. We've got the forearm millimeters. So these are the columns where we have raw data. Okay, all the other columns are what's called transform data. So essentially, I was going to show you how to make a graph, as I mentioned, but I pressed these three dots up at the top, and I have a Samsung Note, and I happened to press this Explore button that I had never pressed before. Okay. And it was truly amazing. Let's see. Oh, whoops, sorry. Of course, now I'm having trouble. I already started the video several times. We want to explore all the data. Here we go. Okay. So now, by pressing Explore, we got a series of data transformations and graphical representations. Okay, some of these are appropriate to look at and some are not. So that's what we're going to do right now. So this is all the raw data in a line graph. And the program must understand what columns are data based on the fact that they're numeric, right? So there's numbers in the cells of the spreadsheet. And then so you can see here that the column names are represented there, and they're all of those columns we looked at that are specifically in millimeters numeric data, okay? But this kind of graph is not appropriate to look at because this is not continuous data. Each individual is separate, so there should not be a line connecting these individuals. Okay, so let's take a look at wingspan and height. Now, here we have an example of an appropriately transformed graph, right? We have on the x-axis here, we have height, okay, and on the y-axis here, we have wingspan. All right, make that look more like a y. There you go. Now, remember I was going on and on about the normal curve. Okay, this idea that we study biological organisms and we make statistics of central tendency like average, mean, range, etc. mode because we expect biological organisms to conform to a general pattern. And so that's what this graph, this lab is helping you to see. Each one of these blue dots is an individual, okay? They are one cell in that data sheet. But when we look at the relationship between these variables, we see that they follow pretty closely what's called a best fit line, okay? And so that line shouldn't necessarily be there because they are individual measurements. They are individual people. But we can see that the relationship between wingspan and height, there's not a lot of variability. There aren't a lot of random people out here outlying this average tendency, this idea of the normal curve in biological organisms. Okay, now let's go here. So here we have the count of the proportion, and I want you to see that here's the normal curve, okay? It's right here, all right? Thinking about this. If you look at these numbers, these are not in order, right? These two should be over here because they're below if you're thinking about it, and these two should be reversed right here. So if this y-axis was presented in the numeric order of the proportion, then it would follow that normal curve. But what I want you to see is that the majority of the individuals did fall right within an average central tendency of a ratio of 1 to 1.75 millimeters when comparing wingspan and height. Okay, moving right along. I'm not sure what's going on with these graphs because we can't see here far enough into the decimal places to know what the difference is. So let's just move right on. All right, so here we have wingspan. This is a histogram of the wingspan. So this is just a count measurement, okay? This is the actual number of individuals that fell within this particular range. And here on the x-axis, the range is from 1,500 millimeters to 2,100 millimeters, and the bin size, each bar, is 200 millimeters, okay? Now we're looking at forearm and wingspan, so different variables. But again, we're seeing this very close adherence to a best fit line. Most biological organisms are meeting this central tendency. Okay, now we're seeing a pie graph of the proportions, and this kind of gives you that idea if we were thinking about the normal curve and the idea that, you know, most of the organisms are going to be in the middle. That would be blue here, okay? And here we have, I'm going to say maybe 60% falling right within the same exact ratio. And then if we add purple and red and yellow, we get closer and closer to that 90% of the individuals, with 5% being out here, 5% being out here. So, adherence to the normal curve and the central tendency of biological organisms. Okay, this is the last graph. This is not an appropriate representation because this, once again, is a line when really we're talking about individuals that are independent measurements. This is not continuous data, okay, in that sense. And so, there you have it. If you wanted to present anything using your phone to help you manipulate the graphs, please do it and explain what you did, because basically we want you to access science at the point that is most comfortable for you. So thank you, and let me know if you have questions.